In this video, we will explore the topics of genetic recombination, horizontal gene transfer, transformation, transduction, bacterial conjugation, and transposons. Genetic information, as we have discussed, can be copied via replication to produce two identical copies that may be passed to daughter cells. Genetic material passed from parent to offspring is referred to as vertical gene transfer. Both prokaryotes and eukaryotes pass their genes to the next generation in this manner. Eukaryotes are able to achieve genetic recombination through sexual reproduction. Genetic recombination refers to the exchange of nucleotide sequences between two daughter molecules. Genetic recombination occurs during meiosis via a process known as crossing over. Crossing over is one of the events that allow meiosis to produce haploid daughter cells that are genetically unique from the parent. Thus, meiosis is the means by which many eukaryotes accomplish genetic diversity. Bacterial cell division is accomplished through a process known as binary fission. Binary fission results in the formation of two genetically identical daughter cells. Thus, bacteria must employ other means to achieve genetic diversity within their populations. Some bacteria have the ability to share genetic material with other bacteria of the same generation through horizontal gene transfer. Horizontal gene transfer typically involves the transfer of plasmids or small chromosomal fragments from a donor cell to a recipient cell. The recipient cell incorporates the donor's DNA into its own chromosome, then becoming a recombinant cell. The donor and the recipient cells do not have to be of the same species for these events to occur. Horizontal gene transfer is rather rare in nature. There are three mechanisms by which DNA can be horizontally transferred. They are transformation, transduction, and conjugation. Transformation is a process by which a recipient cell acquires naked DNA from the extracellular environment. Bacteria capable of acquiring naked DNA from the environment are said to be competent. This means the bacterial cell has special DNA-binding proteins in their cell walls that allow them to bind to DNA in the extracellular environment. The DNA is then transported into the cytoplasm of the cell to be integrated into the bacterial chromosome. Frederick Griffith first described the process in 1928. Griffith examined two different strains of Streptococcus pneumoniae, one that was capable of producing capsules known as the S strain, and another that could not synthesize capsules known as the R strain. Capsule formation is important for this species to be able to avoid host defenses and cause disease. Therefore, S strain Streptococcus bacteria were always observed to cause disease, while the R strain were not able to do so. To summarize the experiment seen in the image, Griffith found that when dead S strain was in the same environment as live R strain, the R strain acquired the genes for capsule formation. The R strain with the newly acquired DNA could synthesize capsules and thereby cause disease in host organisms. Competency has been generated artificially in labs to aid in recombinant DNA technology. Bacteriophages or phages are viruses that parasitize bacterial cells. Bacterial genetic material may be transferred from one bacterial cell to another by a bacteriophage, a process known as transduction. When lytic bacteriophages infect bacterial cells, they bind to the host cell and inject their nucleic acid inside the bacterial cell. The bacterial chromosome is then degraded, allowing the genetic material of the virus to assume control of the cell and direct the bacterial cell to synthesize new viral components. In order for the virus to move on to another host cell, the virus must package its nucleic acid inside viral proteins before being released from the bacterial cell. In some cases, bacterial DNA is packaged inside the viral proteins by accident, producing a defective phage that will not be able to direct the synthesis of new phages. This defective phage, which is the transducing phage, can bind to another bacterial cell after being released from the host cell. The transducing phage will inject bacterial DNA from the previous host cell into the recipient cell. The DNA is then integrated into the bacterial chromosome of the recipient. There are two types of transduction, generalized and specialized. Generalized transduction occurs when random fragments of the host DNA are enclosed by the transducing phage. In specialized transduction, only specific sequences of the host DNA are transferred along with the phage DNA. Specialized transduction is important medically because it is the mechanism by which several species of bacteria have acquired toxins that have made them pathogenic strains. Such bacteria include Corynebacterium diphtheriae, which causes diphtheria, and E. coli 0157H7, a strain of E. coli that produces a toxin that causes bloody, life-threatening diarrhea. A third process by which bacteria can exchange their genetic material horizontally is bacterial conjugation. Bacterial conjugation is a process that requires a structure known as a pilus. Pili are hollow structures composed of protein that are produced by cells that have fertility plasmids. Cells containing fertility plasmids are referred to as F+ cells, while those lacking fertility plasmids are called F- cells. During conjugation, a pilus is formed by the donor cell that extends from the donor cell to make physical contact with an F- recipient cell. A region of the F plasmid known as the origin of transfer then initiates the transfer of a single strand of the plasmid DNA to the recipient cell. The donor cell synthesizes a complementary strand of DNA to the parental strand that it retains, while the recipient cell synthesizes a complementary strand to the new strand of DNA it receives. Now both the donor and the recipient cells contain F plasmids, making both of them F+ cells. The cells involved in this process do not have to be of the same species. The process differs from transformation and transduction in that the donor cell remains alive in conjugation. Conjugation allows bacteria to transfer copies of the genes they encode to new host cells. The genes that are sometimes transferred by conjugation are those for antimicrobial resistance. In some cells, the F plasmid does not remain as a separate entity in the cytoplasm of the bacterial cell, but rather becomes integrated into the chromosome. Bacterial cells in which the F plasmid is integrated in the bacterial chromosome are called high frequency of recombination cells, or HFR cells. During conjugation between an HFR and an F- recipient cell, the HFR chromosome is replicated and a single parental strand of DNA is transferred to the recipient. The F factor separates in the middle, and a portion of the F factor leads the chromosomal genes into the F- cell. Often the chromosome breaks due to the physical separation of the cells, resulting in only a portion of the bacterial chromosome and F factor being transferred to the recipient. The lack of a complete F factor prevents the recipient from being able to produce pili and therefore remains F-. The donated DNA is integrated into the chromosome of the recipient. The plasmids integrate at specific sites within the chromosome, and as a result, the genes are transferred in a specific order. Scientists have used HFR conjugation as a tool to map genes on bacterial chromosomes. Genetic variation can also result from the action of transposons. Transposons, also known as jumping genes, are segments of DNA capable of transposing or moving from one location in the genome to another. The transposon may move to another location on the same DNA molecule or to a different DNA molecule within the genome, and can insert themselves into the DNA sequence almost anywhere in the genome. Some transposons move about within the genome by a cut and paste mechanism, in which the sequence is removed from one site and inserted into another. Other transposons replicate their sequence, leaving a copy of themselves at their original site and insert a copy at another site elsewhere in the genome. Transposons are capable of moving because of the presence of the transposase gene. Simple transposons, known as insertion sequences, are constructed such that the gene for transposase is flanked by palindromic sequences. Palindromic sequences, also known as inverted repeats, are sequences of DNA that are identical to an inverted sequence in the complementary strand. For example, ACTGAT is the palindrome of TAG-TCA. Insertion sequences do not carry genes for any other function. Complex transposons are composed of two insertion sequences flanking genes that are not associated with transposition activity. When the insertion sequences of the transposon are cut from the DNA and moved, they also move the genes located between the insertion sequences. The genes that can be moved include genes like those for antibiotic resistance. Transposons can allow a gene, such as a resistance gene, to be moved from a bacterial chromosome to a plasmid, which can then be shared with other bacterial species, thereby allowing them to spread antibiotic resistance.